Hey guys, this video is going to be about the cell membrane and the transport across it. And this is actually going to be, it's your second PowerPoint for the unit, but it's technically your third set of notes. So just to kind of do a quick recap, so your first set of notes was about cells, and that's where you learned about prokaryotes versus eukaryotes, a little bit about endosymbiotic theory, just basic size of cells, things like that. And hopefully you guys did well on the quiz over that. Your second set of notes is actually not a PowerPoint, but the cell parts lab, where you essentially had to do the matching and learn the organelles and parts and their structures and functions, as well as the diagrams, the endomembrane system, things like that. That is technically considered a set of notes for the unit as well. So I just want you to be aware of that. Now, when it comes to your test, you don't necessarily need to expect to see diagrams, but you will see questions about cell parts and anything that was on that cell parts lab. So this is technically note set number 3, and we're going to split it over a couple days. We could finish it all in one day, but I don't want to overwhelm you. Also, I want to take some time to look at the essay for this particular test because it falls within this content. So we're going to start off with just the basic structure of the cell membrane, and also could be called the plasma membrane. They're the same thing. And this is where we're going to see our macromolecules coming into play, because in our cell membrane we're actually going to see 3 of the 4 macromolecules. Now, the main structure of the membrane, and hopefully you'll remember this from your macromolecule notes, but the main structure of the membrane are these lipids that we call phospholipids. And then there's also going to be some proteins that are big and chunky and have a variety of functions. But the basic structure is going to be what we call a phospholipid bilayer. Now, this is going to go back to the structure of the phospholipid. Okay, the phospholipid is unique because it is amphipathic. Now, if you look at this prefix ampha, think about like an amphibian. It's both land and water. Ampha kind of refers to both. And so in this case, the phospholipids are both hydrophilic and hydrophobic. And this right here, this basic property of these phospholipids is what's going to give rise to the main component of our cell membrane. So a phospholipid, I know I can't draw very well on your slides, but I'm going to do my best. A phospholipid, just to kind of simply draw it, looks something like this. So on one end we have what's called the head, and the head of the phospholipid is hydrophilic. It likes water. And then we have 2 of these tails. Now, these tails are actually long fatty acids. These are fatty acids that could be saturated or unsaturated. They're essentially long hydrocarbons. There's a long tail of carbons surrounded by hydrogens, and these are the region of the molecule that do not like water. So when we're talking about the phospholipids being amphipathic, it's referring to the hydrophilic water-loving heads and the hydrophobic water-fearing tails. Now, something that you need to understand is that cells are aqueous. Okay, living cells are going to contain water. Think about that cytoplasm or that cytosol. It's just a giant solution filled with water and solutes. So the cytoplasm is going to be a solution, but then you're also going to have the outside of the cell, or the extracellular environment, and cells are going to be within a liquid. Even in our bodies, the cells are going to be bathed in body fluids, and so cells are aqueous. They're going to have water in them, and they're also going to have water outside of them. And so the basic structure of our membrane comes from this amphipathic quality of our phospholipids. And so what will happen is, if you've got a cell, remember that you've got water on the inside and you've got water on the outside. And so what's going to happen is that these tails here do not like water. They're going to repel it. And so the tails are going to turn inward like this, and the heads are going to go on the outside of the membrane. And so that way, where you have water inside the cell and water outside the cell, these heads are facing the water and the tails are turned away. So it's due to those hydrophobic qualities of the tails that cause them to turn inward. And so what we get is a basic phospholipid bilayer. So this is your main structure of the membrane, is this hydrophilic heads on the outside and hydrophobic tail structure on the inside. And that way the tails are protected from the aqueous solutions, and the heads, which like water, are touching them. Now, the structure itself, these phospholipids, they're not very rigid, okay? They're not bonded tightly together. And so when you see these heads kind of lined up together, they're not tightly bound. So it's kind of a flexible type of relationship. In fact, they can actually move around on that side of the membrane. It's kind of a dynamic structure, and that these phospholipids can move around. Now, they can't jump across, but with on the same side, they can kind of move and shift because they aren't tightly bonded to each other. And so it is a fluid type of structure. So like I mentioned, they can move laterally, they can stay with on their side. Now, in addition to that, in the inside, remember you have saturated versus unsaturated fatty acids. And so saturated fatty acids are kind of straight like this. Unsaturated had those kinks. And so if you have unsaturated fatty acids, it's just going to increase that fluidity of the structure. Now, I will say though that the membrane is not just one fluid blob of goo, okay? It does have some other structures that are going to make it a bit more stable. And one of those things are going to be proteins. So the main component of the membrane are your phospholipids. The second major component are going to be chunky proteins. Now, I want you to go back to macromolecules. Proteins always take on a very specific shape, which means they do a very specific job. And there's going to be multiple types of proteins within our cell membrane, but they are always going to be specific, okay? Now, two general categories of proteins that you can see in the membrane are your peripheral and your integral. And so if you have your membrane here, you got your heads on the outside, you got your tails on the inside, you kind of have this basic structure going on. When you're talking about your proteins, a peripheral protein just kind of sits on the side. So it's going to be on one side or the other of the membrane. A lot of your peripheral proteins may be things like receptors, possibly enzymes. Now, an integral protein is actually going to go all the way through the membrane. It is integrated. And so these integral proteins are often going to be things like channels or pumps. A lot of your integral proteins are going to be used for transport purposes. Now, we know that proteins have different qualities depending on the amino acids that make them up. Some proteins are hydrophobic, some proteins are hydrophilic, so it just depends on the type of protein and its structure. And so like I mentioned, there's a lot of different types of proteins that you can find in the membrane. Now, channel proteins and carrier proteins, both of these are transport proteins. Channel proteins just have little holes in them and it allows molecules to move on through. Carrier proteins usually have some kind of interaction that has to happen before that a molecule can move through. There's also proteins that we're going to call pumps, which are going to be active transport proteins, but there's lots of different transporters. Now, your receptors are going to be receiving messages. When we get to a lesson called cell communication, you're going to learn that these receptors are incredibly specific. But when you think about things like hormones, for instance, or neurotransmitters, we have these chemical signals inside of our bodies, and these chemical signals, in order to communicate or talk to the cells, the way they do that is they bind to a protein receptor. And so receptors are just going to receive messages or receive signals or chemical messengers. We also have some proteins that help in cell recognition. Like on our blood cells, your blood type may be O positive or your blood type may be AB positive or something like that. That has to do with proteins on our cells, and so proteins kind of act as kind of name tags in some ways. It's how we can recognize our cells as either self versus non-self. We can recognize the proteins as being part of us or not part of us, and so proteins can help in kind of cell communication that way as well. And then, of course, some proteins can be enzymes, which those are just going to be proteins that regulate chemical reactions and kind of regulate our metabolism. So there's all different types of proteins. And not only are they going to have these functions, but they're also going to contribute to the overall structure of the membrane. So kind of the most common accepted model of the membrane is what we call the fluid mosaic model. Fluid because it's not rigid. It does have some flexibility to it, but it's also called a mosaic because there's many different components. So you can see that there's a lot going on. Okay, the basic are these little grape looking things. There's my hydrophilic heads, all of that. Now keep in mind this is a 3D structure, so we always kind of draw it 2D like a flat cell. But there's my basic phospholipid heads, and then inside you can see those hydrophobic tails. You got your big chunky proteins. This would be an integral protein. This right here would be a peripheral protein. Now notice there's a couple other things that are part of this membrane. So here we have another lipid, cholesterol, and I'm going to go back and show you in the picture. I want you to notice where cholesterol is. Okay, cholesterol is a lipid, which means it does not like water. And I want you to notice that all of these little cholesterol molecules are in that hydrophobic region. They're going to be where those tails are because they don't like water. But cholesterol is a type of steroid. That's why I kind of had that little 4-ring structure. But cholesterol helps to stabilize. It helps it make the membrane a little less fluid and kind of stiffens the membrane. It also lowers the temperature, like the freezing point for our membranes, which is good because we wouldn't want our membranes freezing up. And then the last kind of component of the membrane is going to be carbohydrates. And so typically they're oligosaccharides. Now you really don't have to know what this means, but when we did our macromolecules, we had monosaccharides, disaccharides, and polysaccharides. We didn't really talk about oligo, but these oligo are between a di and a poly. They're more than 2, but they're probably not like hundreds like your typical polysaccharide. They may be like, 20, 15, 30, something like that. So they're kind of shorter chains. They're not enormous chains of carbohydrates, but they're kind of bigger than a disaccharide. And so what these carbohydrates do is they kind of help in that cell-to-cell recognition, kind of like some of the proteins. They kind of act as ID kind of labels for the cell and help cells communicate and identify each other. And so I want to go back because there's actually 2 types of carbohydrates. So you can see these little sugar chains here. Okay, notice they're not super long, but they're not tiny either. And so this particular carbohydrate is attached to a protein. And so we would call this a glycoprotein. So the glycoprotein is going to be one that is attached to a carb. So glyco refers to sugar, and then the protein, of course, is the protein. Now over here, like here's one, and then here's one, you can see that these carbohydrate chains are attached to the actual phospholipid bilayer. So we would call these glycolipids. So if the sugars are attached to a protein, they're glycoproteins. If the sugars are attached to a fat, they are glycolipids. And so you see how they kind of just stick out here. I will say that the carbs are always outside of the cell. One of the things you hopefully saw in your cell parts was the extracellular matrix. And so that's going to be all of this stuff that sticks off of the cell. And so your carbohydrates are part of that extracellular matrix. All right, so as a whole in our cell membrane, we have 3 of our 4 macromolecules. We have our phospholipids, which make the basic structure. We have another lipid called cholesterol, which helps to stabilize. We have big chunky proteins that have a variety of functions. And then we have little sugar chains or carbohydrates that help in identifying cells. And so this very distinct specific structure of the membrane is going to give it an important function. And so the main job of our membranes, it is a barrier, but the main job is it's going to regulate the movement of substances across, going into the cell and out of the cell. And we can describe our membrane as selectively permeable or semi-permeable. I want you to know that these are the same thing, okay? Selectively permeable is one way to say it. Semi-permeable is another way to say it. So the word permeable, if we say that the membrane is permeable, that means that the molecules can pass through. So like the membrane is permeable to water. Water can pass through the cell membrane. And then there's going to be some substances that are what we refer to as impermeable. The membrane is impermeable to really, really big, large substances like a huge carbohydrate, for instance. And so we describe our membrane as selectively permeable or semi-permeable because that just simply means that it's picky. Some things can pass, but some things cannot, okay? And so it's going to depend on the size of the molecule. It's going to depend on the properties of the molecule. Is it hydrophobic? Is it hydrophilic? Does it have a charge to it? All of those things are going to affect whether or not a molecule can pass. And so it does depend on the lipid bilayer and those hydrophobic areas. It also depends on the proteins. So normally if they're really small and uncharged, they can pass through pretty easily. But if they're large or if they do carry a charge, they may require proteins to pass through. So it just depends on the nature of the molecules that are passing. And so as I mentioned earlier, cells are aqueous and our cytoplasm is a giant solution. And so I don't know that we've actually covered this. We may have touched on it, but in a solution, something we need to understand is you're going to have two parts. I probably should be writing with the highlighter, but you're going to have the solvent, okay, and the solvent is what does the dissolving. This is going to be water. In terms of our cells, it's going to be water. And the other component of that solution is going to be the solute. And so as we start to talk about movement of molecules, we do need to understand the basics kind of components of a solution. So you have your solvent, which is going to be water. It does the dissolving. And then you're going to have your solutes, which are going to be all the things that are dissolved in that water. And so the cytoplasm is a solution. There's lots of different solutes. There's going to be salt. There's going to be sugars. There's going to be ions. There's going to be vitamins. There's going to be monomers, things like amino acids or monosaccharides. So there's all different types of substances that are solutes within that solution of the cytosol. Now the thing is when you have a cell, remember that there's going to be two solutions. There's going to be whatever the solution is inside the cell, and then there's going to be whatever that solution is outside the cell. And so if the concentrations are different inside and outside, that gives us what we call a concentration gradient. So we could be talking about salt, like for instance, inside of our cells, we have a salt concentration of 0.9%. And so if we had a solution outside of our cells that had more or less salt, we now have a concentration gradient. Okay, so it's just a difference in concentration across the membrane. Now, hopefully you remember this from biology. This is something we hit pretty hard. Or maybe hopefully you just know this just from everyday life. But molecules will naturally move from high to low. Think about downhill versus uphill. You don't really have to do a lot to go downhill. And so that is a natural passive movement. Molecules will naturally move from where they are higher to where they are lower. Things like to spread out. And so our first type of transport is going to be just basic diffusion. And diffusion literally, by definition, is the movement of substances from high to low. Now, this doesn't even have to be across the membrane. Perfume molecules diffuse when you spray them in the room, when you have air fresheners, things like that. Diffusion is just the movement from high to low. And so some molecules are going to move across the membrane by just simple diffusion. If they can go across that phospholipid bilayer, they're just going to move on across. Anytime there's a difference in concentration, they're going to move from high to low. So like oxygen can easily diffuse across the cell membrane. Salts can move pretty easily because they're small. And so when these molecules are moving, they are going to move from high to low, and they will continue moving until they reach equilibrium. And so this is going to be the point at which their concentrations are the same. Now, I do want to point out that it says no net movement, okay? This doesn't mean that there's no movement at all. Molecules will still move, but once you reach equilibrium, there will be, let's say this is our cell. Once you reach equilibrium, they'll just kind of move back and forth equally, and they'll kind of maintain that equilibrium. Now, if there's a difference in concentration, those molecules are going to be moving one particular direction. So if they were higher on the outside, they'd be moving in, or vice versa. But once they reach equilibrium, they pretty much kind of move back and forth equally. Another type of transport is going to be osmosis, and this is what your essay is going to be about. Osmosis is the movement of water across the membrane. Now, water is always going to be able to move because it moves through proteins called aquaporins. They are little protein channels that are specific to water. Now, if you think about that membrane, remember you've got those heads on the outside and you've got those tails on the inside. Water can't easily pass through the phospholipids because remember, those tails do not like water. And so what's going to happen is that water can't just go straight through that bilayer. Instead, water has to go through a protein. Okay, so water can pass easily, but it does pass through proteins. And so osmosis is just simply the diffusion of water through that membrane. And so water will move, and something I want you to remember is that salt, or you could just change that to solute, salt sucks. Anytime there is a higher concentration of solute, water is going to be sucked towards it. And so the water is going to move from high to low, and because that membrane is permeable to it, anytime there are concentration differences in our cells, water is going to move, okay? It's always going to move to wherever there is more solute. So just remember that salt sucks or solute sucks, and it's always going to be pulling water in its direction. This is why you don't drink like seawater. This is also why some people put salt on slugs. You're actually sucking all the water out of its body, which is really kind of animal cruelty. I wouldn't recommend that. But in general, salt or solute is going to pull water towards it. And so when we're talking about osmosis and osmotic balance and water movement, remember we always have two solutions, okay? So we're always going to have a solution inside the cell, and we're always going to have a solution outside the cell. So when we're using these terms here, this tonicity, which has to do with concentration, when we're using these terms here, it's always in reference to two solutions. And in this class, in biology, our two solutions are going to be inside the cell and outside the cell. Okay, so these prefixes are going to be kind of important. All right, so if you know the prefixes, that's very helpful. Iso is equal. Hyper is higher. I'm not sure why the highlighter does that. And hypo is lower. Now what you need to understand though is that these prefixes refer to the solute. Okay, so in our solution, we're talking about the solute, which is whatever is dissolved in the water. So it could be salt, it could be sugar, it could be ions. So if it's isotonic, that means the solute concentrations are equal. And so in isotonic conditions, we're going to see kind of that equilibrium state. And what's going to happen is water is going to move back and forth equally. So isotonic is what we want. We want to maintain osmotic balance in our bodies. It's one of those homeostatic criteria that we need. Now hypertonic means higher solute, and if you have more solute, that means you have less water. So keep in mind, these terms describe the solute, but it's the water that is moving. So hypertonic means high solute. Hypotonic means less solute, and if you have less solute, you have more water. And so we're going to take a look at what happens to cells when you put them in these solutions. So what cells want, especially animal cells, is going to be isotonic. Notice it's normal. The cell is going to stay the same. There is movement, but it's going to be back and forth, inside and outside equally. This is what we want for our cells in our bodies, okay? Now I do want to point out it's a little bit different for plant cells. We'll talk about that here in a minute. This is not necessarily the best state for a plant. Flaccid means kind of limp. It's not hurt in any way, but this is not necessarily a perky plant. So we'll look at that here in a minute. Now if you take hypertonic solutions, which are high solute, if you were to place a cell in a hypertonic solution, remember that salt sucks or solute sucks. And so the solute is going to suck the water out of the cell in both plant and animal. And so in animal cells, what's going to happen is the cell is going to shrivel. Sometimes we could also say that the cell crenates. That's a technical term for shriveling. The cell crenates. And you can imagine that we would not want this happening inside of our body. We would not want our cells to be losing water and shriveling up and shrinking. Now in plants, what happens is it does shrivel and shrink, but they have a cell wall that we don't have to worry about. And so what happens is that cell membrane, when it shrivels, see how it kind of pulls away? That's called plasmolyze. Okay, so it's still shriveling, it's still the same basic concept, but it actually has the the pulling away of that cell membrane, which is plasmolysis. Now, hypotonic over here, you may want to remember hypo and hippo, but in a hypotonic solution, there is more solute in the cell, and so water is going to get sucked into the cell. And for an animal cell, it could, possibly take on so much water that it literally bursts or lyses. Lyse means to break. And so in the event that water just keeps going in and keeps going in, it will bust. Otherwise, we could just simply say that it swells up. Now, once again, it's a little bit different with plant cells. Because they have the cell wall here, this right here is actually a happy plant cell. It's what we call turgid for plants. Because in plants, too much water can't go in. What will happen is water will flow in and flow in and flow in, but when it gets full, the cell wall will actually keep it from taking on more water. So where an animal cell might bust, a plant cell actually becomes turgid, which is a firm, perky plant cell. This actually contributes to the structure of plants. That water pushes against that cell wall, and we'll look at that here again in a second. So once again, iso is equal. Cells are going to be normal. Water's going to go back and forth. Hypertonic is high solute. It's going to suck the water out of the cell. And then hypotonic is less solute, so water is going to go into the cell. This is essentially what your essay is going to be, and we'll take a look at that when I get done going over the lesson. All right, so osmosis is incredibly important for cells because anytime there is a concentration difference, water will move. You have to understand that. This is why kidney function is so important. Because if our blood were to become hypertonic or hypotonic, or dehydration is serious, or you could even drink too much water, there's such thing as water poisoning. Anytime our water balance is off, our cells will either be shriveling or swelling, and neither of those is going to be a good thing. So one thing that we living things are going to always have to deal with is osmoregulation. Cells or organisms as a whole are going to have some type of adaptations that help them maintain water balance. Because if water gets out of whack, if those concentrations are not isotonic, then we're going to have shriveling, or we're going to have bursting, and the cells will potentially die, okay? So some of the things that, like, we have these complex excretory systems and urinary systems that help kind of to maintain our water balance. We can also maintain water balance through things like sweating and different processes that occur in our body. Some of your simpler cells, this was in your notes, the contractile vacuole. This is not a very common organelle, but even like some freshwater protists, they have these vacuoles that help pump out excess water. So even your single-celled organisms are going to have some type of osmotic balance. They're going to have ways of regulating that water moving in and out of that cell. Now, as I mentioned earlier, it is a little bit different for plants. Because they have the cell wall, they actually like lots of water going in because it creates turgor pressure. This contributes to the structure of the cell. And so if you think about plants, if you have any plants at home, you may notice that they get kind of droopy, but as soon as you add water, within a short period of time, they perk back up. That's because water is part of their structure. And so when they start to lose water, they just get kind of limp, or what you can say is flaccid. And so if it loses too much water, that plasmolysis can happen, and that's actually going to kill the cell. So too much water being pulled out is going to lead to the death of cells for those plants. But if it's just flaccid, it can be recovered. And once you add water, the water will go in, it will fill up that big central vacuole. It then pushes against the cell wall, and that's what we call that turgor pressure. So for plants, hypotonic isn't so bad because it makes them turgid, but hypertonic could be very dangerous for plant cells too. Now, everything we've covered so far, diffusion, osmosis, both of those transports are what we call passive. So we have two main types of transport. We have passive and active, and we're only going to cover passive today. We'll save active for the next lesson. But passive is anytime something is moving high to low, and so this does not require energy. So diffusion was substances moving high to low. Osmosis was water moving high to low. So anytime you have high to low or that downhill movement, we consider that passive transport. So osmosis is specifically water, but both of these require no energy. You don't need energy to go downhill, okay? Now there's one other type of passive transport that we're going to look at. We've actually kind of talked about it a little bit, but that is facilitated diffusion. So simple diffusion is just high to low through the membrane, like moving through the phospholipid bilayer. If it's facilitated diffusion, this is when it involves a protein. So it's still high to low. It's just when the molecules pass through proteins. So technically osmosis, when water is moving, osmosis is a type of facilitated diffusion because those water molecules are moving through those aquaporin proteins. And so once again, we still have high to low, no energy is required, but substances are just moving through the big chunky proteins. So this is going to be moving through our channel proteins or our carrier proteins. And so if it were just basic diffusion, those molecules would be passing through that bilayer. But if it's facilitated diffusion, they're moving directly through those big proteins. And so the transport proteins are specific. It's not like there's a general protein that lets everything go through. Proteins are always specific to their job. And so it's, there are certain proteins for certain molecules, like aquaporins are specific to water. There's certain gated channels, like there's sodium gated channels, there's calcium gated channels, there's potassium gated channels. They're only going to let certain ions pass through. Some of these are open all of the time, and some of them have to have a stimulus that opens them, or some of them have to have a chemical signal that has to bind to open them. But the main thing is that in facilitated diffusion, you have these carrier proteins that are going to allow molecules to pass through. And this is where we're going to stop for the day, actually. We're going to pick up with active transport in our next lesson. It won't take us too terribly long. But I do want to take a minute to talk about your essay for this test, okay? It is in your study guide. Hopefully you got your study guide the day that you covered your cell notes. And so I wanted to kind of go ahead and pull it up and just talk about this for a moment. So kind of like the last essay, you're going to take your test, and then after you take your test, you're going to get this diagram right here. You're going to fill this in. So it's not a long like sentence essay. It's another chart that you're going to fill out. And it's all about osmosis and those tenicities. Now, if you look in your study guide, I've kind of reworded it here. This is what you're going to see on the test. You may not have this exact same wording on the essay that's in your study guide. I tried to make it simpler and easier to understand. So essentially what you're doing is our blood has a normal salinity. Salinity is salt concentration. Our blood has a normal salinity of about 0.9% salt. Okay, salinity is salt. And so essentially what you're going to do is you're going to say what happens to cells if you were to place them in different concentrations. Okay, so you have three concentrations. You have a 10% salt solution, a 0.87% salt solution, and a 0% salt solution. So over here in this column, let's see, I'm going to move to a pen. Not really a fan of red. We'll do purple. It's my favorite. So over here in this column, this is where you're going to put the percentage. So literally, you're going to be writing these three percentages. It doesn't matter in what order, but you're just going to be doing those percents, okay? So this will be my 0.87. I'm not sure how that happened. Let's try that again. I was trying to draw an 8 and it thought I was drawing a heart. So there's my 0.87, and then I'm going to have my 0. All right, so over here in this column, you're putting the concentration, and what I want you to do here is I want you to identify it as hyper, hypo, or iso. So here's our original, and so you're going to tell me whether 10% is hyper, hypo, or iso to this percentage right here. I'm not going to tell you because I want you to figure that out, but that's what you're going to be putting in this column here. Okay, so there's two parts. You're going to put the percentage, and you're going to put whether it's hyper, hypo, or iso. Okay. Now, in this column, you're going to tell me what happens to the cell. And so this does not need to be complicated, okay? This does not need to be complicated at all. And over here is where you're telling me why, okay? So this is going to be really simple. So when it comes to what happens, you pretty much have three options. the cell is either going to stay the same, the cell is going to swell, or you could say possibly lyse, or the cell is going to shrivel. Now these are animal cells, so we don't have to worry about, you know, the cell wall or turgid or anything like that. If you put animal cells in different solutions, they're either going to stay the same, they're going to swell up and get bigger and possibly lyse, or they're going to shrivel up and lose water, okay? Now, over here is where you're telling me why. This is where you're going to talk about water movement. It says what is happening in terms of osmosis. So this is where you're going to bring in the concepts of the water moving and why it's moving in that direction. So this is going to tie into whether it's hypo, hyper, or iso. So if you have an isotonic solution, one of these things are going to happen. And why? Why does it stay the same, or why does it swell, or why does it shrivel? So this one you're going to talk about water movement. Like you're going to say something like water moves into the cell because there is more solute in the cell, something like that. Or water is going to move out of the cell, or water is going to move equally back and forth. So this is where you're doing the explaining. So this one should have a little bit more information in it. I mean, it's still a chart. You don't need to have a whole page worth. But this is just naming percentages and identifying them as hyper, hypo, or iso. This is telling me what's going to happen to the cell, and this is telling me why that happens. So you're talking about osmosis, which means you're talking about the water movement. Where is the water moving, and why is it moving there? And so that's going to be your essay for this lesson. It'll be on the test only. You don't have a quiz because we're going to be doing a lab over the course of a few days. And so there is no quiz over the cell membrane and transport. But when you take your test, you will see material on the test as well as this essay here. So that's all I have for you today, guys. I hope you have a great day, and I'll hopefully be seeing you soon. I plan on coming by the school and getting those labs ready. So y'all have a wonderful day and a wonderful weekend.